Hey there! What if I told you scientists can now literally edit your DNA and they've already used it to cure a baby from a deadly disease? If you've ever felt like your genes determine your fate, this video is going to blow that wide open. By the end, you'll understand how CRISPR and gene editing could erase deadly diseases or even boost your health for life. Stick around, because if you don't know how this works, you might miss the single biggest medical breakthrough of our time. Alright, before we get into CRISPR and how it saved KJ's life, we gotta take a quick detour into the blueprint of life itself, DNA. Now, don't worry, I'm not gonna throw a big biology textbook at you, this is gonna make sense. So, think of DNA as a massive instruction manual that lives inside every cell in your body. It's written in a special language made up of just 4 letters, A, T, C, and G. These letters aren't random, they are arranged in super specific sequences. Kind of like how letters in our language help us form words, sentences, and eventually entire books. But instead of writing stories, DNA writes you. It tells your body how to make proteins, tiny molecular machines that do everything, from building your skin to digesting your lunch. It's like the code that tells your body how to operate, repair, and grow. Now, here's the thing. Just like a single typo in a recipe can completely mess up your dinner, a single typo in your DNA, called a mutation, can cause serious problems. That's what happened with KJ. He was born with a rare genetic condition called carbamoyl phosphate synthetase 1 deficiency, or CPS1 deficiency for short. Yeah, sounds complicated, but let's break it down. This condition affects an enzyme, CPS1. This enzyme is absolutely essential for something called the urea cycle. Now, the urea cycle is the process your body uses to get rid of excess nitrogen when it breaks down proteins. Normally, nitrogen gets converted into a less toxic compound called urea, and then it exits the body through urine. But without a proper functioning CPS1 enzyme, this doesn't happen. Instead, nitrogen builds up in the blood in the form of ammonia, and trust me, that's bad news. Ammonia is toxic to the brain. In babies like KJ, it can lead to lethargy, vomiting, seizures, coma, and even death. Here's the kicker. This whole life-threatening cascade, it was caused by a single mutation in his DNA. There are over 7,000 known rare disorders, and around 80% of them have a genetic origin. Many of them just as serious, and most caused by just one small mutation. It's mind-blowing to think that your entire life can be affected by a single letter being out of place in your DNA. But that's also what makes CRISPR so powerful, because if that one letter can be the difference between life and death, then fixing it, that's the cure. Now, just to give you a bit more context, CPS1 deficiency is ultra rare. We are talking fewer than 1 in a million births. There's no cure, and management usually involves a low protein diet, medications to remove excess nitrogen, and in the worst cases, a liver transplant. And even then, outcomes aren't guaranteed. So when KJ was diagnosed, the odds were stacked against him. And this is where the CRISPR magic enters the story, but we'll get to that in a second. For now, here's the takeaway. Your DNA is the foundation of everything in your body. When that code goes wrong, especially in genes responsible for critical enzymes like CPS1, the effects can be devastating, but understanding the problem is the first step to solving it. And that's exactly what scientists and doctors did in the KJ's case. They zoom in on the exact point in his DNA where the mutation occurred and decided to fix it, not manage it, fix it. We are talking molecular level precision, and yeah, the tech that made it possible is called CRISPR. Let's dive into that next. So, what is CRISPR and how does it work? You probably heard the term in the news, maybe tied to genetic editing, designer babies, or miracle cures. But what is CRISPR really? Let's break it down in plain terms. CRISPR stands for Cluster Regularly Interspaced Short Palindromic Repeats. Sounds like jargon, but here's the thing. What it does is far more important than what it stands for. CRISPR is a genetic editing system, and not just any system. is like the find and replace function in a word processor, but instead of editing a document, you're editing the genetic code of living cells. But where did this tool come from? Here's the cool part. CRISPR is actually based on a natural defense system found in bacteria. A while back, scientists noticed that bacteria could remember the viruses they've been attacked by before. They did this by storing little snippets of viral DNA in their own genome, in weird repeated patterns that CRISPR is named after. When the virus returned, the bacteria would recognize it and send out a protein called Cas9, a sort of molecule scissors to cut the viral DNA and destroy it. So scientists said, what if we borrow that system? Could we program it to cut human DNA instead? And the answer was yes. That's how we got CRISPR-Cas9, the most widely used gene editing tool in science today. Here's how it works. First, scientists designed a guide RNA, which is a short strand of genetic code that matches the DNA they want to edit. Then, this guide leads the Cas9 protein to the exact spot in the DNA. Cas9 then makes a precise cut in the DNA strand. Finally, the cell's natural repair machinery steps in and fixes the break. And if scientists provide a new piece of DNA at this point, the cell can use it as a template to rewrite the sequence. With this tool, we can delete malfunctioning genes, insert working ones, or make precise edits, all with a level of accuracy that was unthinkable a decade ago. It's powerful, it's versatile, and it's fast. But, and this is the key, CRISPR-Cas isn't perfect. While it can make very precise cuts, the way the DNA repairs itself afterward can sometimes be messy. Imagine snipping a sentence out of a paragraph and trying to tape it back together. It might work fine, or it might leave a few letters out or add some in. That's why newer forms of gene editing are being developed, like base editing, which we'll talk about in the next section. These aim to be more precise, more predictable, and less likely to cause unintended effects. But before we get there, for the first time in history, we are not just reading the code of life, we are editing it. KJ was born in the United States in 2024. At first, he seemed perfectly healthy, but just a few days after birth, he began having seizures. Blood tests revealed dangerous high levels of ammonia. Doctors soon discovered the cause, a rare genetic disorder called CPS1 deficiency. And in the KJ's case, the root of the problem came down to a single letter typo in his DNA. One base in the CPS1 gene had been switched. KJ's parents were told the truth. No approved treatment existed. His condition would likely worsen. But then, a team of researchers and clinicians came together to create a personalized gene therapy just for him. And instead of using a standard CRISPR-Cas9, they reached for something newer, base editing. Base editing is like CRISPR's more elegant cousin. It still uses a guide RNA to find the faulty gene, but instead of cutting the DNA, it chemically converts one base into another, A to G or C to T, without breaking the DNA strand at all. In KJ's case, that meant switching an A back to the G it was supposed to be. Just one change, one letter, one life. And remember, this wasn't just a lab experiment. This was a real baby, a real human patient. So everything had to be tested rigorously in animals and cells first, especially in mice with the same mutation. And it worked. At that point, the team went to the US FDA and got the green light for what's called a single patient investigational new drug application. At just 7 months old, KJ became the first person ever to receive in vivo base editing directly in his body. So, did it work? Well, the full results are still unfolding as doctors monitor his progress, but early signs were promising. Within weeks, his ammonia blood levels began to normalize, his seizures reduced, and he started smiling more, sitting up, and engaging with the world. And while this isn't a guaranteed cure, it's the closest thing KJ's family and the world has seen for a disease that was once a death sentence. That's the power of gene editing. And it sets a precedent. KJ might be the first, but he won't be the last. The technology is still young, but it's advancing fast, and it opens the door to a future where we don't just treat genetic diseases, we fix them, one letter at a time. So now that we've seen what happened with KJ, this tiny human whose life was literally rewritten at the genetic level, let's zoom out and talk about what this means for the rest of us. Let's start with the most obvious implication. In vivo gene editing is no longer science fiction. It's here, it's been done, and it worked. Now that scientists have safely edited cells inside a living human, we're talking about potential cures for dozens and maybe hundreds of genetic diseases that were previously untouchable. But it's not just about rare diseases. For example, a team of researchers from the University of Amsterdam presented not too long ago a proof of concept towards curing HIV using CRISPR technology. But let's not pretend this is plug and play. There are huge questions still ahead. How do we ensure edits are truly safe over time? How do we regulate this across different countries and healthcare systems? Who gets access first and who pays for it? The last point is big. These treatments are likely to be expensive at first, hundreds of thousands, even millions per dose. So we're going to need policies, insurance frameworks, and ethical oversight to make sure this isn't just a miracle for the wealthy few, and to make sure it is highly regulated. Remember in 2018 when a Chinese scientist made the headlines and was later jailed for editing the genes of twin babies to make them resistant to HIV? He used CRISPR to modify embryos, implanted them, and brought them to term, without international consensus, peer review, or proper ethical oversight. It caused a global outcry. Scientists condemned it, and in 2019, he was sentenced to 3 years in prison by Chinese authorities for conducting illegal medical practices. That's why every new gene editing treatment, like the one used for KJ, is surrounded by rigorous review, testing, and debate. We also need to be careful with what we're editing. Fixing life-threatening conditions? Amazing. Editing for traits or enhancements? That's where the bioethics gets complicated. This is not the world of Gattaca yet, but if we're not thoughtful, we could get there. Still, with all that said, this is one of the most transformative moments in biomedical science in the past years. KJ's story isn't just a headline, it's a preview of a world where genetic diseases don't define your future. Treatments are personalized at the molecular level, and medicine isn't just reactive, it's proactive, precise, and permanent. So, what do you think? Did you learn something new about what CRISPR means for the future of medicine? If there's anything you'd like me to dive deeper into, leave it in the comments. And as always, thanks for watching. Until next time.